Good morning, ladies and gentlemen. I'm Arif Masud from the University of Illinois at Urbana-Champaign. It's my pleasure to introduce our plenary speaker today, Professor Ellen Kuhl from Stanford University. Professor Kuhl received her PS and MS from University of Hanover, her PhD from University of Stuttgart, and her habilitation from University of Kaiserslautern. She joined the faculty of the Technical University of Kaiserslautern in 2002, and then moved to Stanford University in 2007. She is currently the Walter B. Reinhold Professor in the School of Engineering and Robert Bosch Chair of Mechanical Engineering at Stanford University. She also holds a joint appointment in the Department of Bioengineering at Stanford. Professor Kuhl's area of expertise is living matter physics, and she has done fundamental work on the design of theoretical and computational models to simulate and predict the behavior of living systems. Professor Kuhl has pioneered new numerical methods for multi-scale modeling of growth and remodeling to investigate how living matter adapts its form and function to changes in mechanical loading. She's working across a spectrum of scales to show this living tissue adaptation can be traced back to the structural alterations at the cellular level. Dr. Kuhl has published more than 200 peer-reviewed journal articles and has edited 2 books at the interface of engineering and medicine. She's a founding member of the Living Heart Project, which is a translational research initiative to revolutionize cardiovascular science. Ellen is the current chair of the US National Committee on Biomechanics and a member elect of the World Congress of Biomechanics. Dr. Kuhl has received numerous awards for her work. She's a fellow of ASME and a fellow of the American Institute for Mechanical and Biological Engineering. She received the Humboldt Research Award in 2016 and the ASME Ted Belichko Applied Mechanics Award in 2021. Before I hand over the floor, let me make a small announcement. As you are watching this talk, if you have a question for Professor Kuhl, please use the Ask Ellen Kuhl tab on the left side of the platform, tab of the platform. We will open the floor for general questions at the end, where I will read out your question and Professor Kuhl will share her comments with you. So without further ado, here is Professor Kuhl providing her insight into the very interesting topic of neurodegeneration and modeling of dementia. Professor Ellen Kuhl.
Thank you, Arif. First of all, I would like to thank Arif and Krishna and Winkam for organizing such an amazing meeting. I've been in all the sessions hopping back and forth since Monday, and it's really been very inspiring. I would never have thought that an online conference can be this interesting. And I know you have organized 3 conferences, as we've been actually joking, last year, and then this year in person, and then this year virtually, and finally we're now getting together to really celebrate this conference. Thank you for putting this together, and also thank you for inviting me. What I would like to share is some work that we have been doing at Stanford most recently, and I have selected this topic because I think it's a very cool application of the tools that we use in computational mechanics, and they don't usually get along with or across this topic. So I was thinking this would be something to encourage people to get into fields that they're usually not comfortable with, and to show you where we can use common things that we are actually, as a community, very good at. This is, of course, not just my own work. It's work of a group of people, and I'd like to acknowledge Amelie Schäfer, who is a PhD student with us at Stanford, Sveva Fornari, who was a visiting master student from EPFL in Lausanne, Matthias Pirling is a postdoc at Stanford with us right now, and he has just accepted a faculty position at TU Delft in the Netherlands. Kevin Linker is a postdoc who has just gone back to Hamburg-Harburg, the university. Johannes Weikenmeier was a postdoc and is now an assistant professor at the New Jersey Institute of Technology. And then Matthias Jucker is a collaborator. He leads up the German Center for Neurodegenerative Disease in Tübingen. And Alain Goureli is a very close friend and collaborator, professor at applied math at Oxford University. So if you read this topic and you followed the news recently, chances are you have come across a very controversial discussion. And that discussion was around a drug that got approved just a couple of weeks ago. That drug is called aducanumab. And it was in the news because there was a lot of controversy around its approval. So what really makes this drug so stand out? This is a drug to treat Alzheimer's disease. It was the first drug in 18 years that was approved to treat Alzheimer's disease. And actually, more importantly, it was the first drug ever to treat the pathology of the disease, which actually means it treats the origin of the disease and not just the symptoms, the outcome of it. And so, of course, the Alzheimer's community was very excited to see this happen. But then there was a lot of pushback from development, from the FDA. A lot of people from the committee that approved it stepped down when the decision was announced. A lot of clinics actually now already announced that they're not going to prescribe it. So if you wonder why, one reason is that the trial went on for about a decade and the results were somewhat controversial. There's also a huge price tag on this. So this would cost $56,000 per person per year. So this is expensive. And a lot of people question whether that's justified and whether it would take away from the development of other drugs. So before I show you how this drug works and how we can model and simulate things to understand this a little better, I would like to give you a 2-minute tutorial into Alzheimer's disease. So you can see Alois Alzheimer here on the top left and his first patient, Auguste Data, who presented to him in 1901. And then she was actually still pretty young and too young to undergo what we actually see in elderly people quite common. They tend to forget things. They tend to not be remembering things. They have a very scribbly handwriting that you can see here when she tried to write her name. And so Alzheimer thought it was very specific. And he asked to actually get access to her brain once she would pass away, which was a couple of years later. He then did what he does on a regular basis. So he's a pathologist. He slices the brain and stains it for particular traces. And he found two things that are very specific to Alzheimer's. So on the bottom left, you see amyloid beta, which is a clumping of protein clusters in the brain. And then on the right, you see tau tangles, which is also very characteristic. And what's interesting is that to this very day, even more than 100 years later, these two things are the critical traces by which we recognize Alzheimer's disease. So fast forward, today we have 50 million people in the world affected by Alzheimer's disease. This number is expected to triple by the middle of the century. And every 3 seconds, one person is diagnosed with Alzheimer's disease. So as you can see from this slide, you probably don't need to read all of it, but you all know this is a very costly disease. You've seen this from the drug, you know it from caretaking. It's very expensive. And even more so, it actually takes a toll on the caregivers. If you have a friend or family member who is affected, you know that this can be quite a burden, not only financially, but also really on your psychological caretaking. So what's the challenge here? The challenge is that we can detect this disease only very late. And this graph tries to illustrate this. Let me first give you background. This graph is a drawing that clinical practitioners would actually share with their patients or with other researchers. It's purely phenomenological. This is how they draw that this disease spreads. And this also kind of gives you an idea that we know actually quite little about it. So this drawing shows the two things that I've already introduced, amyloid beta and tau as red and blue curves. You see they start developing very, very slowly, and then they ramp up, and then they plateau. And so there's this characteristic sigmoid shape that people imagine that these patterns would take. And you first have amyloid, then tau. And then in orange is something that's mechanical. It's a shrinking of the brain that actually changes shape and stresses in the brain. And then after that, there's a period of cognitive impairment in green. And this is actually the first time that people notice that they're affected by this. And this is when they present at the doctor. And the problem actually becomes visible here. So this is 20 years into the disease. And at that point, amyloid beta and tau and shrinking of the brain have already progressed so far that it's difficult to do anything. So the question is, can we understand this better? Can we detect it earlier? And can we correlate these different tracers? Something great happened a couple of years ago, and I would say this actually has contributed massively to our understanding of Alzheimer's disease. Alzheimer's disease is now commonly viewed as a prion-like disease. And you probably haven't heard of prions before. Prions are proteinous infectious particles, and it's a new mechanism in which a protein itself acts as an infectious agent. So you can think of this as a disease that spreads neither by viruses nor by bacteria, but just by the protein itself. Stanley Prusiner in 1997 won the Nobel Prize for discovering this mechanism. And this is actually something you might have heard of as BSE in cattle, SCARP in sheep, Creutzfeldt-Jakob disease in humans, or Koro in Papua New Guinea. And so these diseases propagate by transmitting a misfolded state, and they have 4 characteristics. They are slow, untreatable, progressive, and always fatal. So we know that neurodegenerative diseases in humans are prion-like. So they transfer a misfolded state onto healthy proteins. And this happens in Alzheimer's disease, as I've just shown, for tau and amyloid beta, but also in Parkinson's disease for alpha-synuclein and in ALS for TTP, a protein that's called TDP43. So another thing that is interesting to note is that clinical people try to understand how this propagates across the brain. And these are drawings that are from a review paper of Matthias Jucker, one of our collaborators. It's a very famous paper in the neuroscience community, and it shows that there's a very stereotypical pattern by which these diseases spread across the brain. So on the left, you can see where they initiate in the brain, all in different regions, and from there they spread across the brain. And then eventually going from left to right, they affect the entire brain. So if you ask yourself how these drawings were generated, this is from hundreds of brains of people who were affected with this disease. When they passed away, the brains were sliced up, they were stained, and then actually these patterns were recognized. So there's a very stereotypical progression of the disease, if you want. So now we've seen there's a growth of proteins and there's a spreading. And as a computation mechanics community, we can identify this relatively easily as a reaction-diffusion equation, right? That's a reaction that happens locally and then a transport, a diffusion through the brain. And so to understand this, we actually start with the reaction for which we use the kinetics of protein misfolding. So this is the reaction, or in our terms, the source term to the equation. We start with the proteins healthy in blue and a misfolded protein in red. And then what needs to happen is there's recruitment, misfolding, and fragmentation. So the healthy protein connects to the misfolded protein, sits on it, it misfolds, and then the 2 fragments. So you have 2 misfolded proteins. So if you're very accurate, you would model all these processes individually. For us, at this purpose, it's sufficient to just model them collectively as a conversion from healthy to diseased. And so we have one parameter, K12, that tells us how fast this transmission takes place. And we can embed this in a slightly more complex kinetics that you can see here. So there's actually production of healthy proteins, clearance of healthy and diseased proteins, and conversion from healthy to diseased. All of these 4 kinetic parameters are potential targets for drugs. And I'll show you how the drug that we talked about earlier affects them. And there's also diffusion. So if you now look at these 2 equations, they all of a sudden look very familiar. There's a diffusion equation or reaction diffusion equation for the healthy and for the misfolded state. We have a diffusion tensor. So this is very common to what we usually do. Our 2 unknowns are P, the healthy concentration, and P tilde, the misfolded concentration. And to make our lives a little simpler, we can assume that there's a huge amount of healthy and a small amount of misfolded protein. So we do a Taylor series around the healthy state. We end up with an equation just for the amount of misfolded protein. So we only have 1 equation here. We can play with this equation and replace some of the constants you see here. We introduce a parameter alpha that summarizes all the kinetics that I've seen before. And then we end up with the equation at the very bottom of the slide, which actually to some of you might be familiar as the Fisher-Kolmogorov equation. This is an equation that's used a lot in applied biomathematics, and it characterizes the spreading and growth of things. It was introduced by Ronald Fisher in a paper in 1937, and it was introduced first to characterize the spreading of mutations of genes in a population of genes. And so this is something that the biology community uses a lot to spread something like that's mutated, in our case, a misfolded protein across something healthy. And it actually works quite well. It's been used broadly in this community and is very popular. So here's the equation on top. The relevant parameters for us are the initial concentration, so that's the seeding of misfolded protein that affects the pattern. And then there's the growth, the parameter alpha, that describes how fast we transition from healthy to diseased. And then there's the spreading. And for the spreading, we can assume it's a second order diffusion tensor. We can assume it has two contributions. One is extracellular, so that's the isotropic spreading. And one is axonal or intracellular. So that's along the direction of the axonal cells. So these are long wires in the brain. And you can think of this as an anisotropic part. We take the equation, and because we're familiar with this, so you could probably all do this, we discretize it with finite differences in time, finite elements in space. We end up with a residual equation that you see on the bottom of the slide. We linearize it for Newton method, and then we can solve it with finite elements. Here are the finite element models that we're using. So on top, you see the model for the brain. It consists of a very high resolution representation of gray and white matter. So gray matter is shown in dark brown here. It's the outer layer of the brain. White matter is the inner core of the brain where the cables connect different regions. And this is the model that Johannes Weikenmeier has created a couple of years ago in our lab. We can then have finer resolution models if we just go to 2 dimensions. So one is a sagittal model that you see on the left, where you cut the brain longitudinally, or the coronal model where you just cut a slice of the brain in this direction. And this runs obviously a little faster, so you can do different trials on these 2-dimensional submodels. We can then seed the proteins, and we do this according to these drawings. So you can see the 2-dimensional model here with the seeding initially in different regions. So let me point towards tau, which is on the top right, for example, where we seed blue in the region of the brain stem, somewhere underneath this hole, which is called the ventricle. And from there, we let it all spread. So we just run the simulation, the reaction-diffusion equation. And this is what you see for these 4 different cases. So you can see the seeding on the left, time progression towards the right, and then you see this pattern develop. And if you compare it to the patterns that I've shown initially, you can see it captures this patterning quite well. And it represents very well what we see qualitatively in these brains. You can also do this in 3 dimensions. So you can see that here. This is the 3-dimensional model, a lot more expensive, obviously, but gives you more information about the depth of the brain. And then you can see on left and right the patterns, how they compare to the drawings. So now, if you recall, we had this sigmoid shape that represents the accumulation of these misfolded proteins in the brain. And it was just a drawing that clinical people assume. And at this point, I would just tell, when I give this talk to clinical collaborators, I would just say, what if you counted the number of colored elements? So for you in our computation mechanics community, this translates into, what if we integrate the amounts of protein that are misfolded across the brain? If we do that over time, we actually recapture this characteristic sigmoid shape. And you can see the equation on the top. So we're integrating the concentration of misfolded protein for every element, so weighted by the size of the element. And then we do this over time. And you can see this characteristic features where for 10 years you have nothing, for steep increase, and then it plateaus at about 20 years into the disease when actually the brain is fully captured with neurodegeneration. So this is reassuring. You can do this also in 3 dimensions and gives you confidence that actually we can capture this curve and we can actually mechanistically explain it, what it really means. It's not just a random curve that we're drawing. So now, being mechanics people, we said, okay, how does this really affect the mechanics? So far, this has been, in a way, chemistry, right, or biochemistry. So how does this translate into atrophy? Atrophy means shrinking of the brain. So how do the red and the blue curve translate into this orange curve? For that, and there we were very lucky because we've been working a lot on volume changes in biological systems. We have worked on growing systems, and this is in a way a shrinking system, but we can use the same and just actually flip in a way the sign of what we're doing. So we do inelasticity for large deformations, which involves a multiplicative decomposition of the deformation gradient that you can see here into an elastic and inelastic part. The inelastic part is the shrinking of the brain, and it captures the atrophy. In this case, it's purely volumetric. So we have a volumetric shrinking parameter theta here. And then the elasticity tensor is essentially just a shrinking scaled deformation tensor. And then from that, we can extract the stresses, purely function of the elastic deformation, and evaluate equilibrium, just very standard what we usually do. You can then actually shrink the brain, and there's two different simulations I'm going to show you. This is called general atrophy. So this is where gray and white matter both shrink, but they shrink at different rates. So gray shrinks a little more than white. And you can see this here, how it evolves over time. You can see the deformations in gray and white matter up to 8 millimeters in the brain. So that actually quite some deformations that associate with this pattern. And you can also see a marked widening of these valleys in the brain. They're called sulci. And that actually is a classical hallmark if you look at the scan of the brain. You can also have focal atrophy, where you just have gray matter shrinking or white matter shrinking. You can see the deformations on top, the phonesis stress at the bottom. And what actually stands out is that there is a clear jump in stresses and deformation at the boundary between gray and white matter. Again, for us, that's quite clear because they're two different materials. But when you talk to clinical people, this is actually where lesions develop. So that's an interesting side aspect of the simulation with mechanics. When you compare this against the features really of shrinking brains, you can capture all of them with a simulation, a widening of these sulci, of these valleys, an enlargement of the ventricle, which is the fluid-filled cavity in the center of the brain, a thinning of the cortex, which is the gray matter layer that surrounds the brain, and a shrinking of the hippocampus, which is this region that's pointed out here at the bottom. So we can couple this, and this is just to show you that we can do a coupled simulation of chemistry and mechanics. And the first row shows the chemics equation that you've seen. The fourth row shows the mechanics equation. And the second and third row in between show the coupling. So we have a deformation-dependent protein spreading and a concentration-dependent shrinkage of the brain. So that's a fully coupled model. And this actually illustrates itself nicely when you do the residuals and you linearize them. You have a full tangent matrix where these two purple or pink terms actually show the deformation-dependent spreading and the concentration-dependent shrinkage. So the off-diagonal terms in the tangent matrix illustrate the coupling quite nicely. So this is what it would look like. This is a simulation that Johannes did in our lab a while ago. On top, you see the spreading of the biomarker, the biochemistry across the brain in years 0, 1, 2, and 3. And then in the middle, you see a scan of a patient who has Alzheimer's disease, where you see these features evolve in time. And then at the very bottom, you see how the brain really shrinks when it's affected by this condition. Then we thought, okay, can we actually explain this by data, large-scale data? And this is a curve fit that actually Amelie did. So you can see, let me walk you through this. On the left, you can see this tau increase in orange over a period of 15 years. And then she calculated the shrinking of the brain. And there's actually two parts to it. So if we want this or not, all our brains are shrinking, and they are shrinking by 1% every year. And this is what this dashed red line shows. On top of that, we overlay the shrinking that's only characteristic for Alzheimer's patients. So that's this dotted line. So if we add both up, we end with this red solid line that actually drastically declines over a period of 15 years. And that shows how the brain is shrinking. You would think that's quite large, but if you look at the right, this is really fitting this to early onset Alzheimer's patients. So this is 40 dots that represent the brains of early onset Alzheimer's disease patients. And you can see how they shrink with onset of the condition and fit actually well onto this red line that I've just shown. So with this, I would like to show you a trick how we actually make this simulation faster. I've shown that we actually use about 500,000 elements when we do a three-dimensional finite element simulation. This is doable. It's not completely undoable, but for the purpose that we have in mind, really running this over multiple brains, identifying parameters. This is, first of all, very expensive, but also we don't really have a lot of information to validate at that level of accuracy. You probably know when you take a brain scan, you will never really get 500,000 points out of this. So what we've done then is we've come across the community in electrophysiology and electrical signaling of the brain, and they actually use a lot networks of the brain. So we started network modeling. And you can see this on the left of this slide. So this is the brain represented as a network. And you can think of this as a connectivity network, how information is propagated across the brain. And the huge red dots in this network are the hubs of information. So these are points where the information passes through a lot. And we can get these points by actually taking an MRI scan. And this has been available through the Human Connectome project. And there are actually 418 images of healthy adult brains. So we averaged over all these brains and got the average network. And what we got is the connectivity weighted network that you see on the left bottom. And if you think of this, it has 83 regions. So these are represented through these dots. And it has all these connectivities in between. And you can think of this as an 83 by 83 matrix. This matrix in network theory is called the adjacency matrix. It has 83 rows and 83 columns, and that's what you see here. So the blue regions on this matrix, these two squares, are where the left and the right hemisphere would communicate, and that's pretty much nowhere. And then the diagonals are very populated, and one of these squares is the left hemisphere, the other is the right, so they look pretty similar. And there are 4 red squares in every one of them, which are the 4 lobes of the brain, and these lobes are closely interconnected. So there also you can see some tight and loose connections. So it's quite insightful to look at this matrix. We use this to actually have a diffusion that's pronounced along this adjacency matrix or this network. So anisotropic diffusion along the connectome defined by image scans of the brain. On the top, you can again see now this drawing, in the middle, the finite element simulation, and at the bottom, you see the connectome simulation. And you can see it can capture the same effects that we have actually seen before. The disease starts in the brain stem. From there, it goes to the front of the brain, to the top, and then to the bottom. And we can actually do this at a fraction of the computational cost. To just show you the result, when we want to validate this, this is a nice way to illustrate that these two methods are quite similar. The continuum simulation that I've shown initially is a dashed black line. The network simulation is a gray dashed line on top of it. So they really reproduce the same result. On top, you see the discrete formulation now solved on the network. And this takes about a second or two. And the continuum simulation with finite elements takes on the order of 1 or 2 days. So it's really a lot more expensive. You can also, for instance, then say, okay, how are these 4 different lobes, these 4 connected regions of the brain, affected over time? So you can see here color-coded these 4 lobes. You can see the disease starts in the temporal lobe, so that's at the bottom of the brain, and then it goes to the front, the frontal lobe, to the top, the parietal lobe, and then to the back of the brain, which is called the occipital lobe. So you see this time sequence in this 4 sigmoid color-coded curves here quite nicely. So this allows us actually to play with this model a lot more. Now the simulations are cheap. They run in MATLAB in a second or 2. And so we can make it more expensive on the local level where we looked at the kinetics. So I would like to go back and really enhance the kinetics and make that a little more comprehensive. And that allows us then to model the effect of drugs, as I will show in a second. So really what is happening at the brain level is we have a buildup of proteins, of clusters. And this happens successively, and we can use what's called the Smoloskovsky model for representation of this. So not only do we produce misfolded proteins of length 2, but they can have any length. And actually only when they become very big is when they clump and when they become dangerous. So what we do is we store proteins of every length, 1, 2, 3, up to the length of 100 or 200, whatever you want. And that gives you a matrix that for every node stores 100 protein lengths, however many we have of those. And so, of course, then the system blows up. So it's, in this case, an 8,300 times 8,300 system. And you can easily see if you do this on 500,000 nodes for finite element mesh, and you store 100 or 200 lengths per node, this can become quite kind of cumbersome. So here are the equations for the Smoloskovsky model. They are very much the same as before. They look a little more complicated. Again, you have this turnover from a shorter to a longer protein, which we call aggregation. And then this other term here is fragmentation. So the 2 parameters are A and F. You can make them longer and shorter. And again, you have all these different kinetic parameters that you can study and see how they dynamically affect the length of these proteins. So now comes a drug. So here's the drug for Alzheimer's disease. And what it does is it clears proteins of a certain length. While it's not entirely correct, it actually tags these proteins. It's called aducanumab, and MAB stands for molecular antibody. And it means it tags to the proteins, and then the body clears the proteins. But they have to have a certain size for this to happen. So let's look at the size distribution. So we plot the same as before. So the dashed line, you've already seen the Fisher-Kolmogorov model, but we can break this line down and see what sizes are present in the brain. So in red, you can see all sizes that are larger than 2, and this exactly fits the Fisher equation. But then you can also say how many of these are larger than 10, 20, 30, 40, in our case, 50. And you can see really the size-specific nucleation, aggregation, and fragmentation dynamics in this graph. So now we can do size-specific clearance with this drug. And for example, we clear proteins that are size 2, 3, 4, 5, or 6, just as an example. And you can see that if this drug targets a specific size, what this would do to the curve, so red is the initial curve, if we now start to clear certain sizes, it would narrow the size distribution. So from red to blue, we're narrowing, and we're also delaying, so we're shifting it to the right. And this is really what we want. So we want to shift this curve as much to the right as possible, meaning we want to delay the onset of the disease. We can't really get rid of it entirely, but if we push it by 20 years, that would be a huge achievement. Right now, the trial is doing this for over a period of 10 years, and it's been showing in these curves that actually it can delay the onset of the disease by several years. You can see this here across the brain. So you can see the sizes 2, 8, and 16 from top to bottom, and their time evolution from left to right. So this is actually quite illustrative to see what would happen if you were to cut off a certain size of protein and you're just left with the small ones. So at this point, I would like to share with you something that actually really has been made possible over the last 3 years. And student Amelie Schäfer was excited to use data of brain scans and actually validate the model. And when she first said, oh, I'm going to do this, I thought this is a massive amount of work. And never, ever will you be able to do this during your PhD. So she has looked at this now for 2.5 years or so, and she's been amazing. And I'm just going to share with you what she has been doing. So a couple of years ago, once we had already started this, it became actually clear that we can also image tau protein in a living brain. And that's been a major change. And this is still developing. And the way this is done is that tracer is induced into the brain, and that tags onto these tau proteins. And that shows under what's called a PET scan, what you see on the left. So some of this lights up on the brain. And that actually means that there's a high concentration of this tau protein. Now we can do this non-invasively, which means we can do this over time and actually really trace the buildup of misfolded protein in the brain. And luckily, this is available. So there's what's called the Alzheimer's Disease Neuroimaging Initiative, ADNI, and they have made all of this available through an NIH-funded research. So when we started this, there were 46 individuals that had more than 2 scans per year, 2 years annual scans. And then 26 of these actually were Alzheimer's patients with an amyloid positive tracing. Amelie took these brains, and you can see the pipeline here, what she did. She registers the PET scan on the MRI scan, which is the structural black and white image that you typically think of when you think of an MRI scan. And then there's a software that's called FreeSurfer that segments the brain into these 83 regions that we use for our network model. And then on each one of these 83 regions, she maps the PET signal and identifies a tau value for each region of interest. And this is the slide that you've seen at the very beginning. So this is doing this for 46 patients in 66 regions. You can see the regions as rows and the patients as columns. And you see multiple bands in one column, and that's the number of years. So for example, you can see 4 columns for four annual scans following up. So the time evolution longitudinally for each patient is shown in each column. Amelie has sorted this to put the most affected regions on the top and the most affected patients to the left. So the top left triangle of this plot is actually showing in kind of red colors. So that's the most affected. And the bottom right is blue. That's the least affected people and regions. So this allows you to actually identify parameters for both the spreading or diffusion kappa and for the production rate alpha. And there's the equation here. this is now solved on the network. You can map the network into the free surfer model, and this is color coding here. So essentially the network simulation maps into regions, and on top you see the mean evolution of tau across all these people, and you can see color coded how it affects the brain. So on the bottom there's a production rate for local growth. It's 0.21, and the global spreading in red, and the parameter for spreading is 0.34. So this is just using a method of least squares, as you can see from the equation on the top. You can use this and do a subject-specific prediction. So you can see the scan on the top of the plot, the model underneath for years 0, 1, and 2. And again, there's a whole brain picture here for the scan and the model in the bottom. And then on the very right, you see the forward prediction, what would happen to the brain according to this model in 10 years. And you can see how this is spreading. So there's a great agreement between the model and the scans, and we can actually predict what would happen in the future to these brains. Now, let me say what I'm actually most excited about in this study is to take this a step further and make it a little more sophisticated. So Amelie has started about half a year ago, and this paper came just out last week, to use Bayesian methods to analyze these data. And this graph here shows the pipeline of this analysis. The top you've already seen, it's a packed images, a segmentation, and the creation of the tau uptake for each patient in each region. The bottom shows how actually this feeds into a classical Bayesian model. So for this Bayesian model, Amelie uses hierarchical priors. And what this allows us to do over the regular just least square fit is we can group and pool the people according to their medical condition, and actually use hierarchical priors for groups and individuals. And then also we can build confidence by looking at the credible intervals of the simulation. So here you see the equation for Bayes' method. We've seen this yesterday in a couple of talks addressed, and we've seen nicely how we can use this in biology. And here we use a likelihood that's normally distributed for the error between the measurement and the model. At the group level, we introduce these parameters that we've seen. So there's the approximation there. And at the subject level, for each subject, we infer a kappa, so that's the diffusion, and then a production. So now what's interesting here is we pool these people into people who show a positive amyloid beta, beta plus, a negative amyloid beta, and we see how that affects the tau. So there's a hypothesis saying, okay, if these groups are significantly different, then actually amyloid beta would be an indicator of tau pathology. So we do this, and you can see the result here on top, the diffusion coefficient and production rate at the group level, and then underneath for each subject. And what we infer is a diffusion coefficient of 1.3 micrometers per year. So that's the mean of the group level simulation. And then you can get the production rates for both groups. So in dark green, the ones that have no trace of amyloid beta, it's minus 0.143. And that means actually that there's more cleared than produced. And then for the amyloid positive, we have more production of tau than clearance. It's plus 0.019. And I'm really excited about this because this actually shows a correlation between amyloid beta and tau, and really shows that there's more clearance in one of these brains and more production in the other group. So this is exciting. And we can do the same as we've done before. We can do a personalized forward simulation. And you can see this here for one region of the brain and for 30-some patients. So this region shown in green is called the entorhinal cortex. And that's a Bragg stage 1 region. And Bragg stage 1 means actually that this is a region that's affected initially by this disease. It's responsible for memory, navigation, and time perception. And this is the first thing that Alzheimer's patients usually are very bad at and lose. And that's when they notice that something is wrong with their brain. You can see the sigmoid shape that we've talked about all along. You can also nicely see a symmetry between the left and right hemispheres, the halves of the brain. The right is plotted as a solid line, the left as a dash. They lie on top of each other very nicely. And for a lot of patients, you see a very narrow credible interval. So that's the 95th percentile of the credible interval shown here as the shaded region. And when the shaded region is narrow, it actually means the model fits these patients very well and the parameters explain the data very well. So if we move forward in time, a region that's slightly later affected is the Bragg stage 4 region. So that happens later. It's called the middle temporal gyrus. And it's responsible for face recognition and emotional recognition. So this happens later in the disease. And that actually we're also familiar with. So Alzheimer's patients have a hard time recognizing people and emotions. And you can see that the curves have now shifted on the graph on this time scale a little bit to the right. And if we move further in time, the supratemporal gyrus becomes affected as a Bragg stage 5, and it's responsible for processing sounds and language. So that's very late in the disease, but Alzheimer's patients have a real hard time recognizing what we say and making sense of that. There's a vertical line, that's a dashed line that Amelie has added to these plots. And that's a line that indicates when the credible interval exceeds 20%. And that's not present in all the plots. So the plots that have a very narrow credible interval are actually showing a good agreement. And in some, she has added this line to actually make a recommendation and say, this would be a good time point for this patient to come back for a scan to actually be highly informative to the model. They don't need to come back any point in time, but this is a good time and we need to rescan. So this is how we can actually inform diagnostics and inform decision-making and identify a time that would be useful to actually do something. So let me close with just a couple of things. We've seen how this drug actually could work. It can potentially shift the amyloid beta curve, and that would affect the tau curve and eventually the shrinking of the brain. So the take home here is, I guess, for all of you to now have a good understanding what the drug would do. We don't have a good quantitative understanding, and since you're all computational mechanics people, can all help to join this force really to understand this better and use your models to kind of predict these outcomes. It's really, models are relatively simple to us, and it can make a huge splash here and actually help a lot to explain this. In terms of the things that I've shown, there's 4 things that I'd like you to somehow take home. So we've shown that a universal law that follows the basic principles of nonlinear reaction and anisotropic diffusion explains how these misfolded proteins in degenerative diseases grow and spread across the brain. We've also shown that physics-based modeling can correctly predict these biomarker curves, these sigmoid curves, and predict the deposition of tau inclusions and amyloid beta and shrinking of the brain. I've tried to convince you that network modeling is actually a good idea, not always, but for this particular model, to predict the spreading across the brain's connectome, where we have a lot of information and scans that we can build into the model. And last and not least, I think Bayesian learning is a very powerful tool that we can use to actually infer model parameters of longitudinal tau PET scans. We can estimate credible intervals and convince people that the model is actually useful. We can do personalized timeline of individual brains or groups of brains, and we can estimate the disease progression under the effect of drugs. Very last, for our community, I would like to reflect on the things that I've heard throughout this conference and put this talk into perspective of this general theme of the conference. So we're all computational mechanics people. I would say it's fair to say we're very good at solving ODEs and PDEs, and we're also very good at correlating physical scales and physical fields. Today, we are in a time where the biomedical sciences are collecting a ton of data at various scales, various devices, various fields. So if you just think about genetics, imaging, or wearable devices, there's just massive data coming into play. So historically, all these people who are collecting the data don't really talk to one another. So if you think of a person, a biologist, they're just measuring something, genetic information in a cell, but they don't talk really to a doctor who treats a patient and vice versa. So these informations are available, but they're not connected. Over the last couple of years, really computer science has taken the stage, and they have offered machine learning as a tool to actually analyze these huge amounts of data. And so I'd like to make the pitch here that that's very powerful, for instance, to analyze radiology images, where it's one field, one thing we want to image, and you've seen this in cancer identification, and it works really well. But in problems that are more complex, that involve different fields, different scales, different different evolutions over time. For example, what I've tried to show, where we have kinetics and we have transport and we have mechanics, this is really neglected entirely. And so that's why these sub-communities have built in our group of computational mechanics people that try to integrate big data, as I've tried to show, with computational mechanics. And we can do this and have a huge impact on human health. For example, we've seen in this conference a lot of mini symposia and talks focusing on physics-based learning or physics-informed neural networks. And I would really like to encourage everybody on this talk to join these forces and actually look at data. There's a ton of data available. You can just be creative, use the tools that we already have to try to help the biomedical sciences and improve human health. Thank you.
Thank you, Ellen, for a very clear and a very comprehensive talk on a very important issue of neurodegeneration. You presented continuum-based models, mechanics models. You talked about network models, and I really liked your reflections for this community where computational mechanics can benefit from machine learning and big data science. So thank you very much for an excellent talk. Now I'll open the floor for general questions. And again, for the audience, if you have any question, please type in the chat box. I will read this question for you, and then Ellen will answer those questions. Let me start off with a question that I may ask. This is regarding the kinetic model. You presented the diffusion reaction equation, and you said it's a prion type problem where folded protein gets converted into unfolded protein, or P converts into P prime. This was in one of your first slides. So let's say, given this finite amount of protein in the brain, so it means the net P plus P prime should be conserved, because P converts into P prime, and P prime is the protein that causes neurodegeneration and associated with all the issues. So my question is, in the kinetic model, do you have constraints and inequalities also that P converts into P prime, not other way around? And net P plus P prime would be conserved?
Yes, so this is a very simple model initially that we use to just understand it. And you're right, there's only a one-way conversion. So you can only produce healthy protein. I think people agree that that's the case. And you can only convert a healthy to a misfolded protein. And that's also, I think, somehow in agreement with what happens. So protein, once they're misfolded, they can't fold back to the healthy state, but they can clear. And that's where the drugs actually attack. So a drug would clear the misfolded state. And then there's, I think what we have to understand, because that's very common to biological systems, equilibrium doesn't mean nothing is happening, right? Equilibrium means there's as much produced as there's cleared. So in a biological equilibrium, things are constantly in motion, but there's a homeostatic equilibrium between what's produced and cleared, right? And so you will always produce, you always clear, but if this comes in out of balance, that's actually when we have more misfolded protein.
Ellen, can you share your screen? I think as you toggle back, maybe audience may not be able to see the screen at the moment.
Yeah, that's... I'm happy to actually share this with people. So if someone just sends me a text, I think this is done. This is handled
online so that they can see.
Yeah.
Okay. So now it's shared on the screen so they can see it. I'll go to the questions. Here is a question from Glacio Polino. Ellen, if I would select one word to review your outstanding lecture, I would say it's inspirational. Fascinating how you connected mechanics and network with Alzheimer. Then the question is, since folding is the domain of origami math, I wonder if origami principles could be used to understand protein folding, misfolding, and their consequences.
I actually think this, so thanks, Glacio, this is a great comment. These proteins, I think, fold it very locally. So I think this might be very comparable to origami, where you have a structure that really folds by very small changes at local hinges. So I think that's probably something that could be used. And you probably know that there's a long unmovable parts in these proteins, which would correspond to your strong rigid parts, of solid parts, and then there's hinges. So people actually use robotics models to model these proteins, where they have a rigid arm and then a linkage that folds. So I think that would be actually a great application. Yeah, great point.
Thanks. Here's another question, and this is from Wei Chen. Yeah, from Wei Chen. Alan, thanks for a great talk. The individualized modeling using Bayesian approach is very interesting. I'm curious how you captured the chemistry difference between different people. Is that an important factor that impacts the disease growth?
Yeah, I think that's a great point. You can see the difference in the different, if we can get the slide up. So this is the slide that shows the differences in the people. So underneath you can see the subject level, and on top you see the group level inference. And so you can see that there's quite some variation between the different people, which I think is common. And that's actually very common to biological data, that you have huge variations. If the group is big enough, I think you get statistical significance. In this case, the green light and dark green curves don't overlap so much, so that actually shows that there is a difference. There is actually, if you look at individual people and you would just compare the subject level, I think you would have a hard time to see whether this is a significant difference or not. So that's a great point. And actually, initially, when we started this, there were just a few data sets available, and we were really frustrated because we didn't see a whole lot. But I think there's now more and more, by huge imaging initiative, more and more made available. So we can use all of this actually. And the more we do this, and you all know this, the more stable actually the inference becomes. That's just the potential of Bayesian inference. The more data you add, you can actually add this gradually, the more it will actually become stable and believable.
Thank you, Ellen. And we are coming to the top of the hour here. So I'd like to thank you once again for an excellent talk and sharing with us your insight into the very important health issue of neurodegeneration. So with that, thank you. And with that, we come to the end of this plenary talk.
Thank you, Arif. Thank you.